Thank you very much for the kind introduction. I'm Koumura from NEDO. Yes. It is a great responsibility that is assigned to me. Thank you very much for giving me this opportunity. I would like to thank the organizers for inviting me today. Earlier, Dr. Toyoda asked me to speak today, and to be honest, I wasn't sure if I should accept the invitation. Since I have been away from molecular machine research for about 2 decades, and I was wondering what I should be talking about today. But since Professor Feringa will be coming, I felt definitely I must be here myself as well. It is held in Sendai, and I am a graduate of Tohoku University myself. So I have many memories in the city of Sendai. And Professor Feringa is here, and my boss, Professor Harada, is also here today. So it is making me very nervous. In front of two bosses, to speak in front of two bosses, it's a very unusual opportunity, but I will do my best. And the title is Introduction to the World of Molecular Machines. Looking at the members of the audience, I see young people as well as senior people. And as far as age groups are concerned, it's quite distributed, but my slides were intended for junior and senior high school student as audience in mind. So I will be covering some basic materials. What is molecular machine? This is from Wikipedia. Molecules or molecular complex controlled at micro or nanoscale to produce mechanical movement, according to Wikipedia. What is important is that it is controlled to produce mechanical movement. Molecular machines are not living organisms, but they move according to the roles given to them. So the movement is controlled. There are roughly 2 categories of molecular machines. One is a biological molecular machines inside bodies of living organisms. Within the living organisms, there are numerous types of proteins. You all have proteins inside your body to maintain life. And proteins play specific roles. And there are many molecular machines. For example, ATP synthase, or myosin, and kinesin, and dynein. So at the center of this slide, you see a diagram of a cell. In senior high school textbook for biology, you might find a diagram like this. There's a nucleus, there's mitochondria in the cell, and there's a lot of space in between, but it's not empty space. And in this space, there are many, many molecules. And that has come to be understood in the past 20 to 30 years. There was a recent TV program by public broadcaster NHK, Human Body, and it's a 3rd installment. If you haven't watched that program, I would strongly recommend that you watch. It's quite interesting. And various molecular machines are also introduced in this program, so I highly recommend this program. And kinesin, dynein, these are examples. And this is a linear motor type of machine. And there's ATP synthase. This is within a mitochondria to produce energy. And this rotates. And so such molecular machines are found in large number in cells. On the other hand, there are artificial molecular machines. Another category is synthesized molecular machines. To put it simply, it uses organic synthesis. In chemistry, we learn organic synthesis methods, and such methods are used to artificially create molecular machines or molecules. And these molecules are given light or heat or electricity or chemical energy as external stimuli. And when stimuli are given, movement is controlled and shape is altered. And if possible, the molecule, as a result, should do some work, some kind of work externally. And such molecules or collective molecules are called artificial molecular machines. And if possible, it says that it should do some kind of work externally. It is becoming possible to control the movement of molecular machines, but how to transfer the force to outside, it's still difficult and challenging. Molecular machines do not have a clear definition. Artificial molecular machines do not have clear definition yet, but I think there are about 3 conditions. Clear direction given a certain stimulus, and here it said state A and state B. It is possible to go back and forth between 2 or more different status, and movement can be observed. These 3 conditions, I think, can define artificial molecular machines. Movement to be observed, observable movement. This last point is very important. Recently, there is a very active research on molecular machines. In comparison to the past, observational technologies are also advancing, and the movement that is controlled is better understood these days. That is an important factor. Now, I would like to get into the details, and I hope I will not lose the audience. In 2016, a Nobel Prize in Chemistry was given to the contribution in designing and synthesizing molecular machines. Professor Feringa and 2 others received Nobel Prize in Chemistry. So I would like to delve deeper into this. Dr. Sauvage and Sir Stoddard, they developed catenan compounds or rotakisan molecules. Using these molecules, molecular muscles were created. There are 2 molecules intertwined, and they contract or extend given stimulus from outside. Such molecules were created, and this part, there are 3 rings, and with external stimulus, it can be moved up and down like an elevator. Such molecular elevator was developed. And as for Professor Feringa, he developed a molecule that move in unidirection, single direction. This is the molecular motor, and let me offer some more detailed commentary on molecular motor. Rotary movement by molecules, and this is a molecular model. Rather than double bond, if there is a single bond, we can easily imagine that it will be rotating quickly, but with a double bond, there is no rotation. But if a light is shown, one of the double bonds will be cut and it will rotate, and this is a trans body and this is cis body. So after half rotation, it turns into a cis body, and such rotation can be caused by light. But the direction of the rotation, if r is smaller in size, the direction of rotation is not fixed, and it may rotate any way freely. So just removing one of the bonds is not a molecular machine. Let's think about a more complex structure. These molecules, this is a trans body and this is cis body, and in the middle there is a double bond, and a certain light is given to the double bond part, and it changes from trans to cis or cis to trans. It isomerizes. This is similar to the top pattern. Light rotates the double bond, but when molecules are as complex as these, rotation should be in only one direction. Both sides, there are naphthalene rings. These are large rings, and in the molecule, it can be causing crowding, and it can be steric hindrance. So there should be only movement in one direction, but it is not observable. So it is not. It cannot be called a molecular machine. So we can add another idea, a rotary movement of molecules. And in a similar manner, we have similar molecules, but there's something that is done in addition in a clever way. Substituent is added as an accent. And let's say this double bond at the center, if a light is given, then it isomerizes to cis body. And in between, from cis body, it can return back to trans body. But instead, this cis body can be given heat. And with heat, naphthalene ring will flip to a different direction and will isomerize in a different direction. So from this cis body, it does not return to the original trans body, but will become a different cis body. And this cis body and newly created cis body and the original trans body, it is not possible to freely go back and forth. So this is a unidirection, single direction, moving only into this new cis body direction. And this newly created cis body, if it is given light, then it will isomerize to trans body and rotate. But it is a trans body that is different from the original trans body, and if heat is given, then it turns into the original trans body. And there is a stationary state between cis body and trans body, and it can go back and forth, but with heat, it is no longer able to go back and forth and will go back only in one direction to original trans body. And with these four different molecules, there is a rotation. On a two-dimensional paper, it is difficult to understand. So let's look at this in a creative way. Blue naphthalene ring. Let's consider this as an axis, and the other naphthalene moves in this way when light is given, and with heat, naphthalene ring will move over to this side.
And as a result of light, it will go in this direction as well, and then it will go back. So as you can see here, the the naphthalene ring, if you look at it from that side, then the naphthalene on the other side will be rotating in one direction. That is how it looks. And with this, finally, we can say that this is molecular motor. It may have been quite complicated, but this is the mechanism. The molecular motors are controlled by light and heat. So up to here, this is the first ever case. It was very exciting, but this was discovered about 25 or 26 years from now, ago. And Dr. Feringa has done a lot of research since then, and let me introduce just one of them. In 2011, this was published, this paper. It is from the Swiss research group that published this, and Dr. Feringa is one of the authors. So this is a molecular car. Here, the molecular motor mechanism is in one tire. It's like a molecular car. So this is how the molecules were constructed. And here, by using electrical force, this will rotate. So this is quite interesting. This tire part, the rotational direction, if it is aligned, then it will move in a straight line. But if the direction of the rotation offsets each other, then this will no longer move. Or if in one direction you align the rotation, so this side goes to the right-hand side and this side would rotate to the left-hand side. So if you design the molecules as such, then they don't know where to go. So the rotational direction here is determined, and based on that, this is reflected onto the movement of the molecules overall. This is extremely interesting molecular system. So the principles of the molecular machines were applied as such. 2016, the Nobel Prize for Chemistry was awarded, and the following year in 2017, nanocar race. This is an international race that was conducted in France. I'm sure many of you are aware of this. And Japan's NIMS team also participated. Unfortunately, they had to retire midway. So there was a joint team between US and Austria, and the molecules from this team won by a huge difference. They were overwhelmingly won. So it may be quite difficult to see right in the center. Dr. Feringa's discovery, the molecular motor mechanism is here. So this rotates and it moves up on its substrate. So this is the molecule that was constructed by the US-Austria joint team that won by a large margin. The nano car race. What is the size? Not even 100 nano, I believe. It's almost invisible, very, very small world. That is where the race was conducted. And the second nano car race was also held, but the first one, there was a lot of buzz for that. But the second, not too much. Maybe it was too small to be the center of attention. But by looking at molecules and moving these molecules, now that is possible. The world of molecular machines there's been a lot of active research in this field. And going forward, I'm sure there will be many molecules that will be developed, and molecular machines will be developed artificially as well, and will be applied in many fields. This is my last slide. This is the Groningen University when I was studying there. The right-hand side is the headquarters. In 1614, this university was opened. So 1614 in Japan, there was a lot of fighting in the Osaka area. And even at that time, a university was established. This is the second in the Netherlands. The first one that was opened was Leiden University. This is very famous, I'm sure you know. And the second was Groningen University. So the headquarters is in the center of Groningen city, but the actual campus where the laboratories are and where the students study is located up north at Zernike complex. When I studied there, it was around the year 2000, somewhere around then. So in 2017, I went back for the first time in years and I took some pictures. When I went to Zernike complex, I was very much surprised. Right here, you can see this red and white object or monument. I hope you can see this is an enlargement. This is what it looks like. I couldn't help but take a photo. So the molecular card that I introduced earlier was here. I was very, very surprised. Well, of course, compared to the actual molecule, I don't know how much bigger it is. But with this, I would like to end my talk. Dr. Feringa will follow. He will be talking about something big story concerning very small molecules. I hope we are all looking forward to it. Thank you very much.